Good morning, this is Harsha Choudhary, pursuing my graduation from Dr. Hari Singh Gaur Central University, currently in BSc 6th Sem. Under the guidance of our professor, Dr. Dipali Jat ma'am, I am going to explain genetics in animal improvement, which is my topic. First and foremost, let's know what animal improvement is about. So, animal improvement or breeding involves a selective breeding of domestic animal with the intention to improve desirable and heritable qualities in the next generation. It is about selective breeding and with a predefined goal in mind, which is to genetically improve the population in a certain direction. So the best animals according to the predefined list of traits are selected, and those selected animals are used for breeding the next generation so that the offspring on average will be better than the parents. Thus, as we can see in the slide, that a male and female of the required traits are taken, the fusion of gametes take place, which results into a genetically improved offspring. Mankind started to create breeds accompanied with artificial selection 250 years ago. Nowadays, breeding of high productive farm animals like cattle, pigs and poultry are improved and bred. It aims at the improvement of animals by changing their genetic abilities for important traits. This brings us to the next part of the slide, which is the history. Now, until roughly the 1700s, animal breeding did not really exist. Of course, people mated their animals with animals in the neighborhood that they liked. There was no systematic way of selecting animals for reproduction based on the predefined characteristics that did not change from mating to mating. Then, in 1859, Charles Darwin published his book on the origin of species based on the findings that he collected during his voyage on the Beagle. He discovered the forces of natural selection. Now, most of the animal breeding theory that we are still using today was invented in the first half of the 20th century. A lot happened in the 20th century with its influence on animal breeding. And lastly, father of animal breeding is J.L. Lush. With this, we come to the introduction of DNA in breeding. Although animal breeding was practiced long before the science of genetics and the relevant disciplines of population and quantitative genetics were known, breeding programs have mainly relied on simply selecting and mating the best individuals on their own or relatives' performance. This is based on sound quantitative genetic principles. Now, until 1953, scientists used statistics and presumed mechanisms to make predictions about inheritance. Nobody knew what exactly was the mechanism behind it. Since the discovery of the DNA structure, a lot has happened. Genetic improvement implies change. For a change to be an improvement, the overall effects of the change must bring positive benefits to the owners of the animal in question or to the owner's community. Moreover, to be an improvement, the effects of the change should bring positive benefits in both the short and the long term, or at minimum, a short-term benefit should not result in long-term harm. The various development techniques involved are: first, we'll discuss about genetic engineering. Genetic engineering, also called genetic modification, is the direct manipulation of an organism's genome using biotechnology. Now, genetic engineering alters the genetic makeup of an organism using techniques that remove heritable material or that introduce DNA prepared outside the organism either directly into the host or into a cell that is then fused or hybridized with the host. This involves using recombinant nucleic acid, which is DNA or RNA, techniques to form new combination of heritable genetic material, followed by the incorporation of that material either indirectly through a vector system or directly through microinjection, macroinjection and microencapsulation techniques. Second is quantitative genetics, which is a breeding scheme that aims to achieve genetic improvement in the breeding goal through the selection of the animals that will produce the next generation. Lastly, we will talk about molecular genetics. Now, molecular information is increasingly used to assist breed conservation programs and to improve understanding of the origin and domestication of livestock. Molecular genetics involves first, that is gene-based selection, in which the increasing knowledge of the animal's genome increases the prospects for applying this technology and provides new tools with which to select for healthy animals. Next is marker assisted selection. In this, if a gene or locus with a major effect can be identified and if a molecular test can be designed, animals' genotypes at the locus can be used for selection. Now, in other cases, a chromosomal region close to the gene of interest may be identified and used as a marker. Lastly, developments in reproductive technology. Now, reproductive technology has a direct effect on the rate of genetic improvement. It includes artificial insemination. Now, the use of AI results in higher selection intensity, more accurate selection of males based on progeny testing, and more accurate estimation of breeding value across herds. Multiple ovulation and embryo transfer. Increasing the reproductive rate of females by moet is mainly useful in species with low reproductive rates such as cattle. The benefits are higher selection intensity on the female side and more accurate estimation of breeding values. Next, we move forward to the elements required in a breeding program. Now, the elements required in a breeding program depends on the choice of the general breeding strategy. As we can see in the slide, there are three major elements that we will discuss. First and foremost is selection between breeds. It is the substitution of a genetically inferior breed by a superior one. This can be done at once or gradually by repeated backcrossing with the superior breed. A male from a genetically superior breed is taken and a female from a genetically superior breed is taken and mated, which results into a genetically modified offspring. Next is crossbreeding. Crossbreeding is defined as the process or the act of producing offspring, particularly through mating two purebred individuals but come from different breeds, varieties, or even species. So, a purebred male from breed 1 is mated with a purebred female from breed 2. Lastly, is within breed selection. Now it gives the slowest genetic improvement, especially if the generation interval is long. In this, a male from breed 1 and a female from the same breed is mated, which results into the offspring. Gradual genetic improvement is the most sustainable form of improvement. Next, we come to the results or the advantages of animal breeding. Now, selective animal breeding already has almost 300 years of history. A lot has been achieved since then. Obvious results have been achieved. The size of the genetic improvement from generation to generation depends on the technique used to select animals for breeding. Introduction of new selection techniques made it possible to select the best animals for breeding more accurately and efficiently. Genetic engineering of agriculture animals has the potential to improve disease resistance by introducing specific genes into livestock. Then it increases milk and meat opportunity in animal agriculture as it increases the concentration of existing proteins or producing entirely new proteins. The control of the quality, color, yield, and ease of harvest of hair, wool, and fiber for fabric and yarn production has been an area of focus in livestock. Thus, genetics in animal production has a growing number of practical benefits, such as in the production of transgenic animals resist to disease, increasing productivity of animals, in the treatment of genetic disorders, and in the production of vaccines. Now, it is not all positive that has come out of animal breeding practices. There are examples where selective breeding has been taken too far. There are also examples where selective breeding has not only improved certain performances but simultaneously and unintendedly also deteriorated other performances that were not under selection, the so-called negative correlated responses. For example, selection for large offspring has resulted in a high fraction of difficult births in sheep. Then broilers started to show metabolic health problem due to the fast growth. Dairy cows and sows started to show reduced fertility during the high production period. Laying hens started to have increased bone fractures because they couldn't manage sufficient calcium intake to deposit into the increasing number of eggs. Thus, this was all about genetics and animal improvement. Thank you for being a patient audience and giving me your valuable time.